Hi, I'm Jan Witkowski. I am Director of the Banbury Centre here at Cold Spring Harbour Laboratory. And this, we think, is the fourth day of the symposium, but we're now in distorted symposium time. And it's the 79th Cold Spring Harbour Laboratory Symposium on Quantitative Biology. And the topic this year is cognition. I'm delighted to have Eric Nestler from Mount Sinai with me. Eric has just given his talk, and I thought we might really go over your talk again, Eric.
Sure, absolutely.
So one of the problems about using mice in any of these sort of higher level things is having a model that to some degree reflects faithfully what one might expect in humans.
Yes, exactly. Most of the depression field has utilized tests that are called despair tests, something like the forced swim test, where a mouse or rat would be placed in a beaker of water. It would swim for a while, then stop swimming. People say that when it stops swimming, it's despairing, although we really have no reason to think that. When you give the animal an acute dose of an antidepressant, it swims a little longer. And it's hard to imagine a test with such little etiologic validity and face validity being so widely used in depression research. So several years ago, we set out to try to develop a test that is better validated, has more ethological relevance for the mouse. And we figured social stress was a domain of behavior that really hadn't been interrogated very much in the depression field with respect to animal models. So in our test, and this is work initiated by Olivier Berton, who is a postdoc in my lab now at Penn, we took a C57 normal inbred mouse and placed it in the cage of a retired male breeder CD1 mouse. The retired breeder is bigger, meaner, and as soon as the test mouse is placed in that cage, the larger mouse attacks it. We only allow the physical interaction to occur for a couple minutes and then place a screen between the mice. So the test mouse continues to be subjected to all the aggressive cues of the larger mouse, but no more physical contact, and the mouse spends the rest of the day in that context. And then we repeat that process every day, typically for 10 days. And at the end of 10 days of doing that, we can show that we have induced in that C57 mouse a behavioral syndrome that's characterized by anhedonia, which means a loss of interest in pleasurable activities. The mice aren't interested in drinking sugar water, eating Reese's Pieces, having sex, running on a running wheel.
Your mice have a good life, apart from these ones.
Yes, that's right, giving the
Reese's
choice of
Pieces,
those range of
sex,
behaviors. Yes, exactly. The mice also show anxiety-like behaviors. They develop a metabolic syndrome. They actually eat too much and get fat and develop a pre-diabetic state. And they become profoundly socially avoidant. And you might think that after 10 days of being beaten up essentially by different mice, social avoidance is a normal response. But actually we've shown if we take these defeated mice and put them back in normal caging with their brothers with whom they were raised, for the rest of their lives, they remain avoidant and don't play in the cages like their brothers. So we think it's clearly pathologic. The behaviors are corrected by chronic administration of antidepressants that work in human. And interestingly, there's a small subset of C57 mice that don't develop the syndrome, and we call them resilient. And we can show that they've been subjected to the same level of stress, yet somehow they manage to avoid many of these deleterious changes.
And do males and females respond? Oh, it's only males.
This is only a male test.
Right.
We've been very interested in adapting social defeat for females, but it's something that's still in progress.
And do you know anything about the difference between the resilient and the susceptible?
Right now, we have no measure that we can apply to a C57 mouse before the stress that predicts accurately whether they're going to be susceptible or resilient. So it's a latent feature in these animals. After the fact, we have described very large differences in brain and peripheral measures that distinguish resilient and susceptible animals that seem to drive those different states, but we don't know what actually causes it initially.
So how did you get interested in chromatin remodeling in relation to this, well, phenotype, I suppose,
so,
induced phenotype?
So over the years, my lab has been interested in mechanisms by which an environmental stimulus, a drug of abuse, stress, might produce long-lasting changes. Because we know in humans that in addiction syndromes or depression syndromes, environmental experiences really change people for their lifetimes and make them more or less vulnerable to the condition. involved. And so we've been struggling to identify the ways in which an environmental stimulus can change the brain essentially permanently. And about 10 years ago, studies in the developmental biology and cancer biology field turned to epigenetics to show that epigenetic modifications of DNA and histones and other things can cause lifelong changes. And so we basically applied that insight to the brain, asking whether epigenetic modifications could mediate similarly lifelong changes in brain and behavior.
And so, did you do a screen for chromatin or epigenetically related molecules?
Yes, so over the last 5 years, we've screened in many different ways. We've screened different histone modifications. We've screened different chromatin remodeling proteins. Chromatin remodelers are ATPase complexes that control the movement of nucleosomes along a strand of DNA, as well as other mechanisms. And we were particularly struck that when we carried out RNA-seq to look at all the global changes in RNA expression in different brain areas as a consequence of susceptibility or resilience, these genes encoding these chromatin remodeling proteins were among the most highly regulated. And that's why we, in the last couple of years, have focused on this family of proteins.
Right. And in particular, ACF1?
Yes. ACF1 is an accessory subunit of the ISY family of chromatin remodeling proteins. There are several families. ISY includes an ATPase, which is a critical subunit called SNF2H. You have to learn like the alphabet soup here. And SNF2H, as an ATPase, can bind to certain subunits and form mechanisms that activate transcription, or bind to other subunits like ACF1 and form complexes that are repressive and decrease gene expression.
And they're repressed by altering chromatin structure?
That's right. They repress by essentially redistributing nucleosomes along a strand of DNA and condensing them, filling in nucleosomes in regions. Take a gene that's active, means its promoter region is relatively spared of nucleosomes. ACF1 SNF2H inhibitory complexes would basically fill nucleosomes in to that region to suppress that gene.
And the question is, what did you find when you looked in your and I guess you use the resistant ones as a second type of control.
Yes, we always look at the resilient animals since they provide an interesting counterpoint to look at an alternative way an animal adapts or responds to stress. And what we found is that in a brain area called the nucleus accumbens, which is one of the brain's reward regions, that there is an induction of ACF1 and an induction of this ACF1 SNF2H complex. And that occurs only in susceptible animals, not in resilient animals. And importantly, suggesting that it may be a factor in depression, and importantly, when we look at human postmortem brain tissue, focusing on this nucleus accumbens brain area, we can actually show an induction of ACF1 in depressed individuals.
Wow.
This is after death, looking post-mortem, but it validates that the finding from the mouse is relevant to the human situation.
And so the next question then is: so now you've found this difference in the chromatin remodeling between susceptible and resilient mice. What genes is it working on?
Right,
or the complex working on?
Yeah, so the first step is to show directly that the ACF complex is involved functionally in mediating susceptibility. Using viral vectors, and this is all work done by Haosheng's son, who's a graduate student in the lab, we were able to overexpress ACF1 or knock it down, specifically in the nucleus accumbens of an adult mouse. And we were able to show that when we mimicked the effect of susceptibility, we overexpressed ACF1, we made the mice more susceptible. And when we knocked down ACF1, preventing that effect seen in susceptibility, we made the mice actually more resilient. And then we used what's called ChIP-seq, or chromatin immunoprecipitation sequencing, to look genome-wide at all the genes that in susceptible mice show increased binding of this ACF1 SNF2H inhibitory complex. We've identified a dozen or so interesting genes. They've never been implicated before, in stress responses or depression. And so now the next step is to actually interrogate those genes to determine how they act alone or in concert with one another to mediate susceptibility.
And will you do that again by overexpression and knockdown?
Yes, we'll use viral vectors initially and then genetic mutant mice as they're available, and explore how manipulating the activity or levels of these genes in the nucleus accumbens controls resilience and susceptibility.
So, is there evidence that coming back sort of to the epigenetic starting point? Is there what's the genetics of acute or severe depression in human beings? Depression
is considered moderately heritable, about a 40% heritability rate, but it's been very difficult to identify individual genes involved. That means that there's a high discordance among identical twins as well. Probably only about 40% chance if one identical twin is depressed that his or her identical twin will also be depressed.
In that case, you could imagine that even for identical twins growing up in apparently an identical environment, there are in fact environmental differences.
Absolutely.
Maybe after they left home or something.
Right.
But you're bringing up your
and
mice.
People have talked about difference in position in the uterus and many birth factors and things like that.
But your mice are growing up in pretty much
We are using
constant
genetically identical animals. We try to raise them in as identical, rote manner as possible, and yet we see this very large divergence between a susceptible response and a resilient response.
I suppose the classic definition of epigenetic is it's an inherent a change in genetic, no, not in genetic material, a change to essentially gene expression not involving structural changes in DNA that is transgenerational.
Exactly.
So the acid test, can you demonstrate such a phenomenon in your mind?
Well, so we're very interested in that, and it's a funny story. We were at UT Southwestern at the time, and two people in my lab, David Dietz and Quincy LaPlante, did an experiment without telling me. And I love when that happens.
Yeah, well, they're always the best.
And what they did was to take male mice that were subjected to social defeat stress, waited 2 weeks, bred them to normal females, removed the males as soon as the females were impregnated, allowed the mothers to raise their pups to term, and then we studied the offspring when they were adults. And what we found was really quite dramatic, that the offspring of defeated fathers demonstrated much greater susceptibility to stress. They had an anxious-like and anxiety-like phenotype at baseline, and were much more susceptible to chronic social stress. And that was an astounding finding. That gets us into the area of Lamarckian mechanisms, which supposedly have been disproved for a century or more.
That's essentially sort of what epigenetic is in a way, isn't it?
That's right, exactly.
So, and you've presumably done this several times. I mean,
Yes.
This is not wasn't
Yeah,
just
no, it's a
one
very
off.
robust and very consistent response.
And so, have you looked at ACF, the genes affected by ACF1 in the offspring before you stress them?
Not yet. I mean, that would be something. It would be hard to do that before the stress because we'd have to kill the animal and analyze the genes.
Right.
If we wanted to do a subsequent behavioral test. However, if we knew that the sons of defeated fathers, for example, showed this increased inherent vulnerability, we could simply analyze their brains and see what changes exist in their brains. It's something we haven't done yet, but we're very interested in doing.
It might be worth trying. I mean,
absolutely.
Oh well, I think next time we'll talk about that.
Okay, sounds good.
Eric, thank you very much.
Very much.
Thank you.
Thank you. Appreciate it.